Hello everyone. Welcome to my channel. Right. Now, what if disease is not something we merely treat, but something we can edit out of existence? Imagine a world where cancer is corrected before it forms, genetic disorders are erased at birth, aging itself becomes programmable, and this is no longer speculative philosophy, and this is the emergent reality of genomic engineering. And at the center of this revolution lies a tool you may already know, which is CRISPR-Cas9. But what comes next goes far beyond editing DNA. So in this part one, let's talk about beyond DNA editing, the epigenome. We often think of DNA as destiny, but in truth, DNA is only the script. The real question is which parts of the script are read, ignored, or amplified. And this is the domain of the epigenome. Next generation CRISPR systems are now being engineered not to cut DNA, but to control gene expression. They can turn genes on or off, modify chromatin structure, reprogram cellular identity. This means we're no longer editing the text of life. We are editing its interpretation. So if, to give you an example, disease may not require permanent genetic change, just precise regulation. Right, so in this part two, I just want to focus on seeing the individual cell, the end of averages. For decades, medicine has treated patients based on averages, average response, average outcomes, average biology, but no disease is truly average, right? And you enter single cell multi-omics. This approach allows us to analyze genomics, which is DNA, and transcriptomics, which is RNA expression, and proteomics, which is the protein activity, and epigenomics, which is the regulatory state at the level of individual cells. Within a single tumor, we now see drug-resistant cells, aggressive subpopulations, and the third one will be dormant precursors. This transforms diagnosis from "you have cancer" to "here are the exact cellular ecosystems driving your disease." And this is not diagnosis, okay? This is biological mapping at microscopic resolution. Now, in this part, I just want to emphasize on programmable gene circuits, cells that compute. Now, imagine this, a cell that can sense, decide, and act. And this is the idea behind programmable gene circuits. By engineering genetic logic into cells, scientists can create systems that detect disease-specific signals, process multiple biological inputs, trigger precise therapeutic responses. For example, a synthetic circuit could be designed to, if as in cancer marker are present, and inflammation marker be high, then release therapeutic protein. And this is not metaphorical, this is not assumption, this is biological computation. We are transforming cells into living machines. right? And now in this part, I want to elaborate further on digital twins simulating the human body. Now let us step beyond the body into digital reflection. A digital twin is a computational model of an individual's biology built from genomic data, um, clinical history, and real time physiological inputs. It allows us to stimulate disease progression, drug responses, and treatment outcomes before any intervention is applied. Imagine testing a therapy not on the patient, but on their virtual biological replica. So this shifts medicine from trial and error to prediction and optimization. Medicine becomes not just reactive, but computationally foresighted. Now, in this part five, um, we are going to look into the most controversial frontier, germline editing. And now we arrive at the most profound and controversial question. Should we edit not just individuals, but future generations? Can we just edit the whole future generations coming along? And germline editing means altering DNA in a way that is heritable. The implications are staggering, like elimination of genetic diseases, enhancement of human traits, permanent alteration of the human gene pool, but also irreversible consequences, ethical inequality, the risk of designed humans. This is not just a scientific decision, it is a civilizational choice. Just because we can does not mean we should. Now, in this part, we are going to learn lessons from a global turning point. Many of these technologies are accelerated during a defining moment in modern science. During the COVID-19 era, during this time, we witnessed rapid genomic sequencing, mRNA-based therapeutics, global scale bioengineering collaboration. It reveals something critical. And biology is now programmable at speed and scale, right? Okay. um, Now let's look at the paradigm shift from treatment to design. What we are witnessing is not just innovation; it is a redefinition of medicine itself. So I'm gonna segregate into three parts. So the first one will be treating disease from designing health. And the second one is reacting to illness from preventing at origin before it happens, before it arises to escalate it to another level. And the third one is one size fits all to hyper personalized biology. And we are moving toward a future where health is engineered, risk is predicted, biology is designed. And the deeper question here is, what is human? At this point, the science forces us into philosophy. If we can edit genes, program cells, simulate biology, design traits, then we must ask, what remains natural? What defines identity, diversity, humanity itself? Genomic engineering is not just rewriting DNA, it's rewriting the definition of life. So in this tonight's lecture, as a closing, I just want to say this as a visionary ending. Let us return to the central idea. For centuries, we have read the book of life. Now we are beginning to write in it, carefully, powerfully, irreversibly. The question is no longer, can we cure disease? The question is, what kind of future are we chosen to create? Because in this new era, we are not just patients of biology, we are becoming its authors, right? So this will be the end of my tonight's short public lecture. And I see you in my next intriguing topics, videos, and have a blessed night. God bless you bountifully in every aspect of your life. Bye-bye.